Hi everybody, it's Miss Bear, and today we are going to learn about a statistical test that you can use when you're comparing your observed values and your expected values in a given experiment. And we're going to use a genetics example since you guys just finished your genetics unit. We're going to use a new genetics example. But this can be applied to a whole range of statistical tests. It doesn't just apply to genetics. Let's look at an example of a dihybrid cross where we're using two different traits, one for the hair length of a rabbit, these adorable little bunnies here, and one for the hair color. So the short hair gene or allele is going to be dominant to the long hair allele, and the black allele is going to be dominant to the white allele. So if these are purebred rabbits, meaning they are homozygous for both traits, what is the genotype of the first generation? I encourage you guys to pause the video and think about these questions after I state the question, because I'm going to go through it relatively quickly, but this is a good opportunity for you to practice these terms that we've learned in class. Let's go through this one. These are purebred rabbits, meaning they are homozygous for both traits. What is the genotype of the first generation? Well, this generation we term the parent generation. And the characteristics, since they're both homozygous dominant, you're going to have one individual who is homozygous dominant, again, for both traits. And you're going to have another individual who's homozygous recessive for both of these traits. Pretty straightforward. Hopefully you guys are able to figure that one out just from the information you were given. Well, now the question here is, if we're going to be breeding these individuals, we want to know what are the possible gametes of these offspring, or what are the possible gametes of this generation. So if you were to do the standard, what we did in class and what you've seen before in other videos, if you were to combine all of these different gamete possibilities, then you would potentially get four different gamete combinations. So for this particular cross, all of those gamete combinations, all of those sperm and egg combinations are going to give you big A, big B, because that's the only thing that the parent has to pass down. Same is true for the recessive individual who has the characteristics big A or little b. Okay, it's going to be how you determine the gametes of these individuals. But no matter what, because they only have homozygous dominant, or they only have the dominant alleles, they only have one potential option for their gametes. We're going to go into a different example in just a second. So when you crossbreed two individuals that have homozygous, or they're considered what's called true breeding. A true breeding individual is one that is dominant or is homozygous for a particular trait, whether that's dominant or recessive. And when you cross this type of a pairing, what you end up with is a unique phenotypic ratio. And we're going to go back to this idea of phenotypic ratios today. But for this guy here, what you end up with was a 16:0 ratio, or a 1:0 ratio, where you have all of the same exact genotype and phenotype. But we're going to go over an example here where you don't have that. Let's say an unethical breeder, which it's very common for people to unethically breed these animals. Let's say you're an unethical breeder and you want to breed two of the rabbits from the last generation. Well, as you guys know, this would be brother and sister pairing, and that's a very dangerous thing to do, because if you're making a brother and sister pairing, that can cause a lot of mutations, right? So let's say that this is an unethical breeding between two brothers and sisters that are both heterozygous, or brother and sister that are both heterozygous. What are going to be the possible gametes of the F2 generation? Well, in this case, what we've said is the F1 generation here. F1 generation is going to be heterozygous for both traits. Both of these individuals, both of these parents are heterozygous for both traits. Now, if you were to go through and again do the same process that we did for the first generation, for the parent generation, you would find out, okay, and again, I encourage you to pause the video at this point, you would find out that these F1 parents can produce four possible gametes. These F1 parents can produce four possible gametes. They can produce the gametes, let me change my color real quick. They can produce gametes that are big A, big B, big A, little b, big, whoops, little a, big B, and little a, little b. Based on the probability rules that we learned about independent assortment, each of these gamete combinations is going to be equally likely, right? That's saying that this dominant A allele is going to combine with this dominant B allele just as often as the dominant A allele is going to combine with the recessive B allele. That's the rules of independent assortment. Okay, so these would be the gametes of our F1 generation. Now, what would be the possible phenotypes of the F2 generation? Now we're trying to figure out, okay, if these individuals breed, what are the actual phenotypes? What's going to be produced in the F2 generation? Well, what this is leading to is the idea that you can do a dihybrid cross again using these same gametes and determine the potential phenotypes and genotypes for each of these two generations. So let's go ahead and do that. I'm going to go ahead and erase my screen. I'm going to go ahead and erase my screen. And there is our possible F1. That actually should say, yeah, that's an F1 generation. And this, what I'm showing you here, this whole Punnett square, this 16 box Punnett square, that's going to be the F2 generation, right? Now, figuring out these genotypes and phenotypes, you typically won't be asked to do any kind of dihybrid cross on your AP test. You may be asked, and it may be a skill that you definitely want to develop. And I encourage you to at least practice one of these, developing a dihybrid cross, figuring out the possible gametes and doing a dihybrid cross, because it is a valuable exercise. But what we really care about for today's exercise and for today's problem is what is going to be the ratio of the phenotypes. What is going to be the ratio of phenotypes based on this cross? Now, if you look at this cross, there's going to be potentially 4 different phenotypes. There's going to be 4 different phenotypes. You're going to have a short-haired black rabbit. You're going to have a short-haired white rabbit. You're going to have a long-haired white rabbit, or you're going to have a long-haired black rabbit. And what we care about for determining if this is what we would expect to see, we care about the ratio of these phenotypes. Now, normally, the ratio, if you look at this, remember a Punnett square represents the probability that a particular gamete or egg and sperm combination will occur. So let's look at all of the possible ways that you could get a short black offspring. This is going to be the dominant characteristic, right? This is the dominant characteristic. So if I'm going to we'll use the green color for this. This is going to be 2 dominant alleles. Now, if you look at this Punnett square, each of these boxes here that I'm going to circle, this is going to represent the possibility of getting a short black offspring. So if you count all those up together, you're going to have a probability of 9 individuals that are short and black, right? 9 individuals that are short and black. So let me go ahead and write that on the screen. 9 individuals that are short and black. If you do the same thing for the short and white generation, we'll do this one in red. Short and white generation is going to be any individual that is dominant for the first allele, short-haired. When I say short, I'm meaning short-haired, and then recessive for the second allele. So that's going to be these 3 possibilities on our Punnett square here. So if you go through and you determine that, what you basically get is the idea that the short white offspring probability is 3 out of 16 chances. Okay, 3 out of 16 chances. Same is true for the long white rabbit. And I'm going to go through the rest of these a little bit faster, right? For the long white rabbit, you're going to have these 3 probabilities here because those represent the genotypes associated with the long white rabbit phenotypes. And then finally, the last possibility that you have is going to be your long black phenotype. And if you look at our Punnett square, there's only one probability out of 16 that you are going to have a long black offspring. So if you look at this ratio, the way that you figure out this ratio, this is a 9 to 3, 9 to 3 to 3 to 1 ratio. Now, this ratio is important. This ratio is very important because what it tells us is if our offspring, if we are a breeder and we breed our rabbits and they have this phenotypic ratio, we know that this gene is going to have a certain inheritance pattern, and we can breed these offspring with reliability. Okay, let's look at what that would actually mean. And I apologize, the colors are going to change a little bit here, but hopefully it won't be too confusing. Let's take a look at what that's actually going to mean when it comes to numbers. If you have, say, for example, this is a huge number, but let's say, for example, that you have 1,600 rabbits from this particular pairing, which rabbits tend to breed really aggressively, but not quite that aggressively. But let's just say this for a hypothetical, right? If you have 1,600 rabbits, how many of each phenotype would be expected based on the ratio, based on the 9 to 3 to 3 to 1 ratio? Well, if you go through and you do these calculations, or you figure out, okay, based out of 16, right? If you have 1,600 rabbits, the expected ratio is going to be out of 16. Sorry. So how you determine that? You're going to do it like this. You're going to take your total number of rabbits, times whatever the ratio, whatever the phenotypic ratio you would expect, which is 9 out of 16. You're going to do the same thing for the short and white, except now you're going to use the ratio 3 out of 16. Do the same thing for the next ratio, 1,600 times 3 out of 16, and same thing for the next ratio, which is 1,600, which is 1 out of 16. What this is going to end up giving you is the actual observed values you would expect based off of these particular phenotypic ratios and the type of inheritance that you would expect for these ratios. So if you do this calculation, you're going to get 900 for the first phenotype, 300 for the second phenotype, 300 for the third phenotype, and 100 for the fourth phenotype. And that's going to give you the expected ratio you would have with this number of offspring. Okay? Let's say, for example, though, that you're the breeder and you're trying to figure out, is this rabbit going to be breeding, or are these rabbits, what is going to be the probability of the offspring that I'm going to get in a particular breeding, right? And you want to figure out, is this what I should expect to see, right? In your pairing, you got these observed frequencies, right? These are the observed frequencies that you actually got when you went and you did this cross, right? You bred 1,600 rabbits, which God knows why you're going to do 1,600 rabbit breedings, or why you're going to have those 1,600 rabbits, right? Now, what we want to know is, are these values close enough to our expected values? Because remember, we calculated that our expected values are going to be 900, 300, 300, and 100. So what we want to know is, are the observed frequencies that we see of these phenotypes equivalent to what we would expect to see? And if they aren't, is there some sort of non-Mendelian genetics contributing to the inheritance of these genes? Because if there is, it's going to be a lot more difficult to breed these rabbits. So this is where the chi-squared value comes in. This is how we do the chi-squared test. Don't get intimidated by the equation. It's actually really easy to use, okay? This equation is going to tell us if there is a difference between our observed and our expected results. So let's crack into it. There's a Bozeman video on this that is excellent, so I encourage you, if this is at all confusing, come back to the Bozeman video and see if you can compare what I say in this video to what he says in his video, because he goes over a lot of the same concepts, just in actually a little bit simpler terms. So here's our expected frequencies, and here's our observed frequencies for each of the following phenotypes. Now, in order to do a chi-squared test, you need to have what's called a null hypothesis. And the null hypothesis is always going to be, for a chi-squared test, your null hypothesis is always that there is no significant difference between the observed and the expected frequencies. This is basically a statement that you are going to use your statistical value, the calculations that you do over the course of a chi-squared test. You're going to use those calculations to decide if you support or reject your null hypothesis. And we represent the null hypothesis with this HO symbol. So if you see this, this is talking about the null hypothesis. Now, you can also have what's called H1. Now, these two are basically saying the opposite of each other. You're typically only going to see H naught is how this is described, or your null hypothesis, which is our observed values are not statistically different from our expected values, which if you're breeding rabbits, this is great. You don't want your statistical observed values to be different from your expected values, because that means that the inheritance pattern is straight up Mendelian genetics, and it's really simple. If you end up supporting, or if you end up rejecting the null hypothesis, that's how you talk about rejecting or accepting a hypothesis. If you end up rejecting your null hypothesis, that means that there is an outside influence affecting the ratios of the phenotypes. So again, this is hypothetical right now, but let's crack into the actual equation. There's going to be, I think, 7 steps that I've kind of boiled down a chi-squared test into. Again, you guys can feel free to pause and take notes if you want, but you can also just do the calculations along with me, right? What you're going to do is you're going to input the observed and the expected values into your chi-squared equation. Now, I haven't gone over any of the pieces of this equation yet, but the question you want to be asking yourself is, okay, we have 4 different observed and 4 different expected frequencies of phenotypes. So which observed and expected ratio do we use? Do we use the ratio for the short black rabbits? Do we use the ratio for the short white rabbits? Which ratios are we going to use? And this is why the chi-squared test can be so nifty, because you're going to use all of these values. You are going to use all of these values. That's what this symbol here, I'm going to circle this symbol in purple. That's what this symbol means. When you do your chi-squared test, this symbol means the sum of something, which in this case, you're going to be taking the sum of all of your expected versus observed frequencies. So that means you're going to do this calculation, observed minus expected squared, over your expected values. And you're going to do this for your first phenotype. So you're going to do this for phenotype number 1. Phenotype number 1. You're going to do the same equation. So you're going to do plus observed minus expected squared over expected. You're going to do that for phenotype number 2. So this is phenotype number 2. And you're going to do that for all of the following phenotypes. So that means you're going to continue to do this equation for however many expected or observed phenotypes that you have, right? So this equation, you put a little dot, dot, dot here. This is saying that you're going to take the sum of all of those equations put together. So remember, your phenotypes here, this is going to be your observed and expected first phenotype, the one that I just highlighted in yellow. And then the next observed and expected ratio is going to be the one that I just highlighted in red, right? So you're going to plug these numbers in multiple times. Now, there's a really easy way, a really nifty way to do this. And you're going to see this on your practice questions as well. There's going to be a practice assignment where you practice doing the chi-squared questions. And that's this diagram here. These are the same observed and expected values that we had in our last diagram. I've just transferred them to a new diagram. So I've basically inputted all of the phenotypes here, and we are going to calculate the following values. Okay, we're going to calculate the following values. Now, I would ask you to do this for yourself. Pause the video, again, pause the video here, and determine these values for yourself. What's going to be the observed minus the expected? What's going to be the observed minus the expected squared? And then what is going to be, let me move my little picture here. What is going to be the observed minus the expected over the expected? And then you add each of these values together. You're going to add each of these different phenotypes together, and that's going to give you your chi-squared value. That's essentially the value that you're trying to calculate using the equation. Okay, make sure you pause the video and try to make this happen, okay? You're going to take your observed minus your expected, which in this case, your observed is 907 minus 900. That's going to give you a observed minus expected squared. That's going to give you, first of all, this is going to give you a 7. That means your observed minus expected squared is going to be 49, which when you calculate that value you're going to end up with, and I don't know that number off the top of my head, so let's do this on a calculator like the astute AP Bio students that we are. So that's 49 divided by 907, which is, if we put it on our table, it's going to be this value that we have here, right? It's going to be this value that you have here. And you're going to do the same thing for all of the following observed and expected ratios. What this is going to eventually give you is when you add all of this column together here, you add all of this column together, you're going to get your chi-squared value, right? I encourage you to come to office hours if you weren't sure how I got these calculations, okay? Now, what do we do with this value? Now that we have determined the sum of all of the expected and observed calculations, what do we actually do with this value? And that's the real difficult part of the chi-squared test that kids often get hung up on. You're going to use the following 4 values that are used. These are what we use to determine, is our observed ratio what we expected to see? Is it close enough to what we expected to see? There are 4 values here that we're going to go over. And the first value is going to be the degrees of freedom for our scenario. So you need to figure out how many degrees of freedom are in our particular example. So the degrees of freedom represents the number of potential outcomes minus 1. So the question I have for you here is, how many different expected and observed outcomes are possible for this calculation? Another way to say this, how many possible phenotypes are there? Think about that for a second. How many possible phenotypes are there? We wrote in the last chart there are 4 potentially different phenotypes, 4 different combinations of these 2 genes. So that means in order to figure out our degrees of freedom, we're going to do 4 potential outcomes, 4 potential phenotypes minus 1, which is equal to 3 potential phenotypes, or 3 degrees of freedom, excuse me. So we're going to go ahead and add that to our box at the top. 3 degrees of freedom. There we go. Now the next value that we are going to use is our alpha value. Now the alpha value is typically something that will be given to you. It represents the probability that our test provides a false support for our null hypothesis, or a false refute of our null hypothesis. So this is basically saying, how accurate do you want your test to be? How much do you want to be sure that your test gave you the right answer? For AP Bio, assume that this value is always going to be 0.5. For AP Bio, assume that this value is always going to be 0.5. This represents confidence. And these are some typical values you might see. They may ask you to use a different alpha value, but these are what these values mean. They basically represent the probability that our test gives us the wrong result. And you're going to typically use the 0.05 value for our tests in this. Whoops. You're going to use the 0.0 value for our tests in this class. So that's going to be this guy here. Sorry, my touchscreen isn't working great. 5% chance that our test gives us the wrong result. This means a 95% chance that we got the right result from our chi-squared test. Okay? That's our alpha value. Now we're going to use the values that we just calculated, the chi-squared, the degrees of freedom, and the alpha value. We're going to use that to figure out what our critical value is, right? Critical value is. So this is what a typical chi-squared table looks like. If you look at the top of this table, the top of this table represents the alpha values. The side of the table represents the degrees of freedom. These are the degrees of freedom, or this little V symbol here. And you go through and use what is determined as your degrees of freedom and your alpha value to determine what your critical value is. So if you go over, remember we said we had degrees of freedom, which are 3. So that means we're going to be in this row right here. And our alpha value is in this row right here. We're going to be using an alpha of 0.05. So what is our critical value? Our critical value is 7.814. 7.814 is our critical value. Now, what do we do with all this information? We've calculated all of these things. What is the point? What do we do with all of this information? You're going to use the critical value that you just calculated, right? You're going to use the critical value that we just calculated, which this is a definition for critical value, but you can come back to. The critical value that we just calculated is what we compare our chi-squared value to. And what you're going to say is your chi-squared value greater or less than our critical value? Is our chi-squared value greater or less than our critical value? So what you're looking at here is you're saying, okay, chi-squared value, this x squared, we use this little x to represent chi, is 0.87 greater or less than 7.81? Well, obviously this is going to be less than 7.81. So what do we do with that information? When you figure out if your chi-squared value is less than or greater than your critical value, this tells you something very important. If your chi-squared is greater, and this says p-value here, that should say critical value. That should say critical value. Sometimes you can use a p-value, but we're using the critical value here. This should say critical value. If your chi-squared value is less than your critical value, you are going to accept the null hypothesis. That null hypothesis that we talked about earlier, we're going to come back to that. If your chi-squared value is greater than the p-value, that means you are going to reject the null hypothesis. Okay? So, based off of this information, based off of what we just calculated, are we going to accept or reject our null hypothesis? Do we accept or reject our null hypothesis? And hopefully you guys can, pause the video and try to figure this out. You are going to accept your null hypothesis. You are going to accept your null hypothesis, which, if you remember, our null hypothesis told us, let me go back to our null hypothesis slide. Our null hypothesis told us that our observed values are not statistically different from our expected values. Our observed values are not statistically different from our expected values. So is there a statistical difference statistically significant difference between the expected and the observed frequencies? Is there a statistically significant difference between the expected and the observed frequencies? The answer is no. For this example that we just did, no. There is no difference between the expected and the observed, statistically significant difference, because obviously there's a difference. There's a difference between 900 and 907. There's a difference between 300 and 289. But we want to know, is this difference statistically significant for us? Okay? Now, why would we use this unless you're a rabbit breeder or a dog breeder? Well, it turns out that Sturtevant and Thomas Morgan did the chi-squared test to determine if his ratio of fruit flies was what he expected. So in this particular breeding, this is a lot of fruit fly genetics, so hopefully this isn't too confusing. But in this particular breeding, Thomas Morgan took two different genes for fruit flies. Gray eyes and normal wings, or black eyes and vestigial wings. So he took these two different traits and he did a Punnett square, did a cross between them. Now, based on the number of flies that he had 2,300 flies. This was the number of flies of each particular phenotype that he should have expected. He should have expected around 575 gray normal winged flies, around 575 gray vestigial winged flies, same for black and normal winged flies, and the same for black vestigial winged flies. This is based off of a cross between the homozygous or the heterozygous individual. These are the gametes, so this means this parent was heterozygous, and then the homozygous recessive. This is not the wild type version. I encourage you to go back and review fruit fly genetics if this is at all confusing, okay? Now, his observed data did not fit the expected results. And so what I encourage you to do is do the chi-squared test for for Thomas Morgan's observed results, figuring out, okay, is this 965 close enough to the 1-to-1-to-1-to-1 ratio we would have expected out of this particular cross? How do the observed values compare to the expected values of this cross? And what he and Morgan and Sturtevant eventually figured out is that this is not statistically close enough to the observed values. These values, 965 and 206, are way off from the expected values that you should have gotten purely based on Mendelian genetics. So what Sturtevant and Morgan had to figure out was why. Why on earth are these observed values so aggressively different? And they came up with a brilliant solution. Sturtevant ended up coming up with a brilliant solution. And that is this idea of linked genes. I encourage you, if you have questions about linked genes and mapping genes on a chromosome, please come to office hours. But if you have questions about this, it's a very interesting topic, because what they figured out was that linked genes are more likely to be found together because they are close together on a chromosome or because they are on the same chromosome at all. They may not always be found together, and this led him to consider the idea that this link between these two characteristics might break occasionally. And these are genes that you know are potentially familiar with, like red hair and green eyes, or blue eyes and blonde hair. Those are potentially linked genes. And this is the idea that Morgan and Sturdivant concluded based on the results of a chi-squared test. They figured out that their observed values were too far away from their expected values to be statistically significant, right? So that's chi-squared test. Hopefully that was helpful for you guys. There's a practice set for you guys to do linked in Schoology, and I will see you guys next time we're in class. Bye, guys.